therefore fertilization happens later, and therefore non-disjunction, the chromosomal abnormalities are going to be much higher in a female embryo compared to a male embryo, which undergoes almost like 30 mitotic divisions before birth. Because there is more number of mitosis, there is going to be a more number of replication errors. So there is going to be a bias in terms of what mutation that gender is going to cause. Then, in a female, it's going to be more of a translocation based. In a male, it's going to be replication errors. So that's exactly what this is talking about here. You see, because the fertilization happens here, and that is why the so-called primary oocyte will now undergo a second meiotic completion of the meiosis. That's where the so-called chromosomal missegregation can happen. And that is why translocation is much greater here. Whereas in this case, spermatogonium continues to replicate here and number of divisions, and that is the reason why the mutational rates are higher here. In a similar fashion, like for example, that's why when a person gets aged and the same spermatogonium is producing a number of times by dividing, and therefore accumulation of mutations at an older age for the male is much, much greater. For instance, that is the reason why even in our laboratory, sometimes if I take an old fish, like we do fish genetics, very old fish, and mate it with a female, I don't need to create ENE mutagenesis at all. We get a lot of mutations simply without having to worry about creating by ENE mutagenesis. So far we talked about basically what types of differences are there in mutations, whether gender plays a role, and nomenclature of these mutations, for example, insertions and deletions as well as point mutations. We need to pay attention to those. And now we're going to talk about genetic diversity in humans. As I pointed out, a lot of them need not be deleterious at all. And those are called neutral mutations. The genetic diversity has been shown since the time they invented the cytogenetics. They looked at staining patterns of chromosomes and they found out that there is a lot of variations in those. And those are copy number variations. You learned about trisomy, the copy number variation. And nucleotide changes, protein changes, all these have been studied before. But there are mutations which will occur in coding sequences as well as non-coding sequences, but they are not going to cause the disease at all. These are mutations still. After all, it is a change in the base pair by your radiation or some other insult. But these mutations which are not at all causing any types of deleterious problems are called polymorphisms. For example, if you take overall polymorphic rate, it is approximately 1 in 1,000 base pairs. You have a change 1 in 1,000 base pairs. Whereas if you take a look at the protein, it is only 1 out of 2,500 base pairs. Why is it that it is present? Protein estimates coding sequences are under selective pressure. So if you have more mutations in protein here, you can have deleterious quickly, right? That is why they are under selective pressure, and therefore there is a variation between estimates from the genome versus estimates from the protein coding sequence. And these are called variations. For example, if I line you up, and look at your patterns, for example, hand patterns of all individuals in the classroom. It's not deleterious mutation, but there is a mutation or a change, and that change led to different variations in your own hand, the palm pattern or something like that. Those are all polymorphisms. So polymorphisms, although it is arising from the genome, you're going to have a phenotypic polymorphism. Like if you take anatomy, a nerve can pass from one direction slightly in one body versus the other. There's a variation. That is not deleterious. That is still a polymorphism. If it is usually 1% is called a variant. If it is less than 1%, it is a rare variant. There are polymorphisms which are deletion polymorphisms, duplication polymorphisms, triplication polymorphisms, and wide varieties, of course. Okay? But they're still polymorphisms because they're not causing a disease. The polymorphisms could be in genes or in introns. And those which cause the disease, some cause the disease, they're called mutations and many do not. But one of the advantages of these polymorphisms, people exploit it, is it is a tool for genetic mapping or gene mapping. Okay. I think we will talk about gene mapping, I think, in the next chapter or so in detail. But right now, the concept is genetic polymorphisms will be helpful in mapping the gene. Suppose if you do not know the location of the gene, it's a new gene that you want to put it back where exactly it is. These polymorphisms will help you to put back this gene in its right place. Polymorphisms have been useful in prenatal diagnosis, blood banking, tissue typing, and organ transplantation. For example, if you want to donate your liver to somebody, they type you, they tissue type you, so that your major histocompatibility antigens are matched. Okay? And that is still based on the polymorphism. They are not going to look at each gene, each protein. The polymorphisms are the ones which are going to help you there. Personalized medicine. That is, nowadays, the new concept is that the whole genome, the whole expression by microarrays we will be able to know in the future. Everybody will be fingerprinted for his or her gene expression profiles. So as we told you, environment plays a significant role. So depending upon the way that the polymorphisms are there in a population, these polymorphisms, let's say a set of polymorphisms, let us say they are associated with increased levels of glucose and decreased levels of glucose. So depending upon overall pattern, you can tell, hey, these individuals are going to be prone for these particular disorders. So that is called personalized medicine. And also a drug dosage. For example, some aspirin, simple aspirin, some individuals may be resistant to it. Some individuals may require less dose or more dose. But if you have this kind of polymorphisms and fingerprinting of all individuals, you can very calculatively tell, hey, look, these individuals may require less dose or more dose based on that. So that's personalized medicine as well. So these are all going to happen in the future. Right now it is not happening. Forensic medicine, forensic application. Okay, you may have an issue with some kind of a crime scene or whatever. So those things can also be detected by polymorphism, by studying the polymorphism. We're going to talk about those in detail as we go along. Paternity issues. Okay. Suppose that the babies are switched or something like that in hospitals. You could possibly, very rarely it happens, but still it can happen. And people have come and really looked at the polymorphic variations and identified whether it's a crime or whatever it is. All these polymorphisms can be studied by looking at what are called a single nucleated polymorphisms called SNPs. And also insertions and deletions, that's what they're called indels. Okay? So mostly these are the two things that they look at in the polymorphisms and connecting to these various types of problems that we have. Single nucleated polymorphisms are 1,000. Just now we talked about 1,000 approximately. Therefore, by definition, because there are 3 billion base pairs, a haploid genome, you're going to have 3 million differences in the human genome between two individuals. Because 1 out of 1,000, so 3 billion divided by 1,000 is a 3 million possible variations. But overall, in all individuals put together in the world, it is only between 2 is 3 million. That doesn't mean you should multiply the 7 billion population. But there are only 10 million possible differences among all individuals. and 10% has been chosen from these mutations for Hapmap. You heard of what is Hapmap? Okay, Hapmap means it is haplotype mapping. Let me give you an example of what is haplotype. Let us say there is a change, that is change 1, R1, R2, R3 and R4. Okay? So that is in one individual. In another individual, it can be R1, R3 and R4. In another one, you may have R1, R2 and R4 and so on. All these possible combinations. So the arrangement of mutations in a linear fashion in one individual could be that, in another individual that one, in another individual that. These are all polymorphisms. That is one haplotype. Okay? Whereas this is another haplotype and that is another haplotype. Okay? So if you have so many 10 million mutations there or whatever, you can have enormous number of possible haplotypes. See, if I take one individual in a given gene locus, the arrangement of those polymorphisms will have one particular arrangement in one individual, one different individuals will have different ones, okay? Because of the so-called innumerable combinations that we can have, the haplotype is going to be very unique for one individual, because the arrangement of the combinations of these so-called polymorphisms will be highly unique, and therefore, if one haplotype is, let us say one particular arrangement is there in one individual, and he commits a crime, there is no way another individual will have an identical haplotype in the 7 billion population. It's very rare, although it can happen. And therefore, you can very easily rule out the possibilities of whether he's going to commit a crime or whether he's going to be related to a particular disease, associated with the disease and so on. So haplotypes will help in associating even an individual with a disorder or individual with a crime or whatever the case may be. So it's a very, very standard, fantastic correlations. Okay? But that doesn't mean haplotype is causing the disease. Haplotypes will never cause a disorder. Haplotype will be only associated with the disorder. Okay? So, and even now, a lot more things are being done with hap mapping because there are so many combinations. What is it? 10 million? You take 10 million, it's actually, even if you take 3 million, out of that only a few percentage of hap map, we're talking about enormous number, and that enormous number with, by variations, you're going to be tremendous hap mapping. We're talking only one locus. I'm not talking about one locus. Imagine the whole genome, and that kind of hap map and association with the disorders is taking time, but people are coming to that and they are doing it. In the future, it will all be done. So, index, insertions and deletions. Half of them are very simple. It only involves 2 alleles. And the other half involves the multi-allelic index. And then there are what are called microsatellites and mini-satellites. The microsatellites are where either it could be a short tandem repeat with a 2 or 3 or 4 nucleotides, whereas mini satellites are having 10 and up to 100 nucleotides. So these are all the numbers. Whereas the copy number polymorphisms, the CNPs, are almost like 200 base pairs to 2 mega base pairs. And these will involve mostly 2 alleles. Okay? And it cannot be possible by cytogenetics because these are too small for cytogenetics. And these are all too large for sequencing. And therefore, the comparative genomic hybridization is the only one by which we can use it for copy number polymorphisms. So, there are various ones. One is microsatellite, one is mini-satellite, and these are also called variable number of tandem repeats or VNTRs, and copy number polymorphisms. All these polymorphisms can be used for mapping studies as well as